Thank you all for having me come give a presentation. I have to confess that I have only been working on this syndrome for the past few years. I had a very interested medical fellow in obstetrics gynecology who came into the lab and was interested in working up some of the cell lines that I had gotten from Lisa Schaefer. So it's been really an exciting project, and I'd love to share today our results with you. And also, some of you probably forgot what you were writing your names and addresses for in the pad that went around in the room. But what I'm interested in doing is really expanding my study and including a lot more families enrolled in trying to really understand how the genes involved in this syndrome could really end up with variable phenotypes, like variable medical problems. In other words, why are some children severely affected with the syndrome and why are some mildly affected? So that's kind of the goal of this study. So here, what we're interested in is, and I'm just looking for the middle red button. I see it. Thank you. So how do we find, so some of this Lisa already mentioned, so how do we find the important genes? And another question is, that we're interested in is, can a piece of the missing chromosome, which is the 1p36 region missing, how can this piece affect the adjacent parts? So in other words, how do you explain why different deletion sizes give the same kind of medical problems? Some children with a smaller deletion are more severely affected than with some with the larger deletion. So why is that? And one question that Lisa posed to me when I first started working on this is, does the deletion affect genes nearby that even aren't deleted on chromosome 1? And really, what are the steps on a molecular level or gene level that go from you're missing one copy of chromosome 1p36, how do you get to the phenotype? So I'm going to go through some basic slides just to remind you and to remind me to tell you that, embryonic development and fetal growth is super complicated. And this depicts the first trimester of embryonic development when the embryo is an embryo, not a fetus. And so there's a lot that has to go on during the first trimester, which starts off with a single cell and ends up with what looks like a person already. So this process is really vulnerable to any kind of genetic or environmental insult. So having a deletion on part of chromosome deletes a whole bunch of genes, and those genes are sensitive to copy number, and that's how you end up having developmental problems. So when you think of an embryo, which is kind of shown in this picture, developing in the uterus, there are different cells in the embryo and different tissues. And so some of the cells contribute on the outside to forming the brain and the nervous system, whereas other tissues participate in forming the muscles and the heart. And other cells contribute to organs like the kidney and the liver. And these are derived from basic cells that are on the outside, the middle, and the inside of an early developing embryo. And all of these affected in individuals with the syndrome, which leads one to think that multiple genes are involved. So I keep using the word genes, and we all using the word genes, but really what are genes? So genes encode proteins, and some of those proteins are important for our bones, for our muscles, for our eyes, for all our body structures. The proteins are the building block of what makes our body. So if there's a problem with the DNA sequence here within a given gene, then RNA is made, which is the DNA gene product to RNA. This gets processed to proteins. So in our lab, we're looking at RNA levels as a way to actually, because it's easier to detect changes than changes in protein levels. So we look at levels of RNA that are encoded by genes that make specific proteins. And these proteins are our building blocks. So, when you think about 1p36, you're thinking about, okay, it's not that you're missing both copies of all the genes in the region. In fact, you're missing one copy out of two. And I kind of drew this scale to depict what is going on. So in the normal situation, this scale would be balanced. But in the situation of the deletion, you have fewer copies, so this thing is going up and down. So you have fewer copies, so you have less of the genes, so the balance is lighter. Whereas if there was a duplication, you'd have too much of genes. So, but very few among, so let's say maybe less than 10%, probably 1 to 5% of genes are sensitive to copy number. So not all of the genes that are deleted on 1p36 are responsible for the syndrome. Probably maybe 10% of those genes. So how do those genes, when combined, lead to the phenotypes? Well, lead to the medical problems. So this is a picture that shows, I think, in a very simple way of some of the slides that Lisa showed. So this is the 1p36 region. This is the very telomere of the chromosome. And I'm not a cytogeneticist, so the bands don't look as nice as on Lisa's slides. And here's the centromere. And this marks all the genes in the region. And these are the different deletion sizes. So as I posed before, why does someone with this size deletion have maybe a more severe phenotype than another child that has this size? And why are there children that have maybe deletions that don't even overlap and yet have the syndrome? And so these are some of the questions that Lisa had presented, makes the syndrome so complicated, but as well makes the syndrome so important to try to understand. Because when you think about it, a lot of these genes are probably mutated or deleted, but just a single gene in just non-syndromic birth defects. So more commonly occurring child with cardiomyopathy or child with intellectual disability. So the genes we find really for this disorder are broadly applicable for many other disorders as well. So when we think about genes and gene products and how you could end up with medical problems, I like to think about it as dominoes. Now, I'm dating myself, but have any of you seen or played with dominoes at all? Oh, awesome. Okay. So dominoes are great. You have to be very patient with dominoes. So you know how you line up all the dominoes, and what is the most fun part? The most fun part is knocking down the first one and watching all of them go down. Well, that's in a way how I think about 1p36, in that you're knocking down a few genes here, and then that just knocks down a whole bunch of other genes or affects a whole bunch of other genes that ends up with the particular medical problems. So our goal is to identify both the genes and all the downstream genes or the additional dominoes so that we could get a better picture, because that might help explain why there is such varied medical problems among individuals. Does that stuff make sense? Yes? Okay, good. So what we do is, since it's impractical to take tissues such as organs, such as the heart from somebody who's affected, obviously, or the brain, so how do we study RNA expression levels and their changes so that we could identify these dominoes? Because what we do is RNA expression levels of genes equal the dominoes. Well, the way we do it is we obtain blood cells. So we were fortunate to obtain transformed cell lines from blood cells from Lisa Schaefer several years ago. And the idea is that even though blood cells are not brain cells and are not heart cells, a lot of genes are expressed in blood cells, and I'm talking about the white blood cells, the lymphocytes. So there are a lot of similarities in genes that are expressed. So if there's, in a way, the blood cell could be a marker or a flag for genes that are affected in the heart or the brain, such that you don't need to look at that tissue. You could kind of look at blood and get some indication that, okay, these are genes that are changed in expression. Maybe those are important genes. So that's what we've been pursuing. And this is what the transformed or you could take a virus and you can infect white blood cells, and then these cells become immortalized. And if you look in a microscope in a culture dish, this is what you see. You see individual cells in a dish that were normal blood cells, but now can grow and grow and grow. And then you see also bunches of cells. And these cells you can culture up, and you could get gigantic amounts of these cells, which is great for genetics research. But our interest now, and the reason why I'm sending the pad around, is because we want to replicate what we found in the cell lines with normal blood samples, because we want to make sure that the changes that we see in RNA expression are not due to the infection of the virus to create this cell culture model, but are actually due to real changes. So what are our interests? So what we want to do is take a blood sample, isolate separately the RNA, which marks gene expression, and the DNA so that we could measure the exact deletion size, and then we could coordinate RNA gene expression with actually present of genes. And what you do is you take the RNA or the DNA and you could put it on this gene chip, which is about half the size of this pointer. And each little gene chip here costs in our lab about $400 to run. So we want to be careful and make sure that we run samples that we feel confident that the child has the deletion, and that we are able to get parental blood samples also to serve as controls. And so in these gene chips here, you can't see it. It looks just like a blue square, but there are thousands and thousands of little probes there that could detect either RNA or DNA level changes. So we want to get an impression of genes changing in expression level in the 1p36 deleted region and outside the deleted region on 1p36 to look for position effects or effects of the deletion on surrounding genes, as well as genes elsewhere in the genome that could explain, again, the differences in medical problems in different unrelated children. So in order to do this, we look at all of the RNA, and then we want to get an overall impression. So the way we get an impression is by sort of a mathematical method in which you create a heat map. So you may wonder, what is a heat map? So here's a heat map of weather in the United States. And so you could get an impression from this heat map. You get an impression because you know what these colors mean. So the hottest is white, then comes red, and the coldest is dark blue. So what you can see is in this part of the country, it's very hot on this given day. And on this part of the country is the coldest part of the country. So that's a heat map. It enables you to immediately get an impression or a feeling about genes. So this is a heat map that we did for RNA levels of all of the genes on the 1p36 deleted region in different individuals. So we had, in this case, we had 7 patients. I'm just counting to be sure. And a whole bunch of different control individuals. And along the bottom are the gene names. So can you get an impression? Well, one thing I'll tell you is that green means the gene is reduced in expression compared to normal, and red means the gene is increased in expression compared to normal. So if you look at this, you see a whole bunch of green. And that makes sense because that's the deleted region. And in the normals, you'd see red as compared to green because it's higher in the normals. So what you're doing is you're comparing one set to the other set. And all of the patients have the same similar size deletion in this particular situation. And then you could kind of start interpreting the results or making diagrams to help explain the results. So this is the centromere of chromosome 1, and then here's going towards the telomere. And this is the 1.36.33 region that Lisa talked about, the 0.32, 0.31. So you're moving back towards the centromere. And again, here is a cartoon of the data that's presented. So what I'm showing you here in this particular individual with the deletion, they have these genes here in green are genes that show reduction in expression. The gray regions show no reduction in expression. So in the lymphoblastoid cell lines, either these genes are not detected or they're not expressed in blood cells, so we can't measure them, or that they're somehow, the deletion on one copy, the normal gene could be expressed more and compensate. What was interesting is that the same genes seem to show this deletion phenotype in different unaffected individuals. Means it's a way to kind of validate that, okay, these genes expression is reduced in general in all individuals here. But what was also interesting is when we looked at the genes outside the deleted interval, we didn't see that same consistent pattern. So we didn't see that there's a whole bunch of genes in this region in all of the individuals with the deletion that show reduction or increased expression. And that gave us the indication that probably this deletion is not affecting the genes or the expression of genes in the surrounding region. And that would be a pretty important finding because, you know, one of the whole big ideas have been is that genes in the deletion region somehow affects expression of surrounding genes. But we must repeat this with more samples to really be sure. Another problem is, you know, when you do use blood samples, again, someone can always question, well, you're not using the brain tissue, you're not using the heart tissue. But on the other hand, it's a good starting point. Let's see. Oh, that's okay. So this is just a picture, this is just a summary, that some of the genes in the deleted region show reduced dosage or reduced expression, and it's consistent between individuals, so it's more trustable, and that there's no consistent changes in gene expression outside the deleted interval, which suggests that probably it's the deletion size itself and the genes within it, plus addition of variations in genes outside the region that explain variable medical problems. So this, again, summarizes our goals. So which genes in the deleted region are reduced in expression? So in the blood cells, only about 30 to 40%. But we hope when we get normal blood that we'll be able to detect more gene expression changes. Oops. Does the deletion alter the chromosome and result in altered gene expression near the deleted region, so outside the deletion? So I just showed that it doesn't seem to be. Now another very important question is, so how do you explain variable medical problems then? So another big question is, what genes elsewhere in the other chromosomes are altered in expression? So as I mentioned, what are the other dominoes? So this is another heat map of the genes only outside the chromosome 1p36. And you could also get an impression. So for example, in all these affected individuals, they share a common set of genes that are upregulated compared to the normal situation. Similarly, they share a whole bunch of genes that are downregulated or expressed lower than compared to normal controls. So then you could start looking at these genes. So, you know, there's so many genes, though, maybe a few hundred that are upregulated in affected individuals or downregulated. How do you make sense out of all this? How do you get a better impression beyond a heat map? So you do this by making another kind of map. So this may look like galaxies, which it's kind of in a way like that. It's a map. But instead of galaxies, these are genes. So this shows a gene network that's important for a particular function. So a whole bunch of genes talk to each other. Like your leg muscle, the muscle cells themselves have a whole bunch of genes and a whole bunch of proteins that are required to form the complicated muscle tissue. And the genes in here are clearly central to the network, whereas the genes out here are probably a little less important. So our interest is really to make these exact network maps and then figure out which are the central genes, and then maybe not worry so much about those genes that are outside. So we've done this. So we looked at all of the genes and we created 3 different networks. One network for neurological development, one network for cardiovascular, and one network for immune system. And these gene names aren't as important, except to note that you can begin to make sense of the data if you use certain computational approaches where you could get an overall impression. So you could get an impression that, yes, there are consistent genes elsewhere in the chromosomes that change in expression due to the deletion. So if we could understand those genes better and their relationship to the genes in the deleted region, then we could better learn how to explain why some children are severely affected and others not. And this is another gene network for cardiovascular development. And what was kind of interesting to me was that some of the genes that seem to be important for cardiovascular development, brain development and function, and immune function are shared. So it gives an impression that there are certain common set of genes that may be affected downstream of the 1p36 region. And this is the immune response genes. So in conclusion of this presentation, we've been using lymphoblastoid cell lines, which lymphoblastoid means tumor-like because we infected them with viruses so they could grow and grow. We use these cell lines to mimic some of the genes that are altered in individuals with monosomy 1p36 syndrome. And we've identified, in fact, some genes and pathways that might help us understand how the deletion leads to the medical problems in individuals. So what is next? So the next goal is to really validate the findings and to expand the study by not having 9 or 10 cell lines from individuals, but having 20 or 30 or more so that we could better understand the basis of this disorder. And the pad that I was passing around is really to kind of get contact information so that I could send you a little bit more information about what we're actually trying to do. And I would like to acknowledge Lisa Schaefer for getting me interested in 1p36 and for our friendship over the years and a great collaboration. And Avi Alkali, who was the fellow in obstetrics and gynecology who did all of the lab work and a lot of the thinking parts, which, you know, along with staying up at night and giving birth and really doing a lot of deliveries of a lot of children and also having 2 children while he was in my lab with his wife. And Dayu Zhang, who is an assistant professor at Einstein, and he's the bioinformatics person who's helping me tremendously, as well as his postdoctoral fellow who did a lot of the work. And thank you for your attention.